That's why I made you a co-host, Carlos. So again, thank you to everyone, because some people might not have heard the welcome. But we're gonna just go into a few little slides I made. Present. Starting with our beautiful illustration made by Malvo. The Home River Bioblitz, the day that we all went out to the river, right? And we all took people along and explored the river. And just seeing from this web of observations on iNaturalist, you can see that it was really like all around the world. People were out in the same day. People were from Chile to India, and from Indonesia to Scotland, from Mexico to Austria. Like, pretty amazing to see that all of those countries were joining. Many observations came in as well, like 80,000, which is, I think, 3 or 4 times more than what we had last year. But mostly what amazed us was just the chat, like the WhatsApp chat that was going on. And it was like active for 24/7 for 3 days long, because people woke up. When I went to bed on the Thursday night, I think, before the Bioblitz, some people in Indonesia were already going out, and that feeling of just this wave going through, this river going through, super cool. In this slide, I just want to give a quick thank you to the whole team, and I want to tell you a bit who was actually behind this. And I took some observations of you all to like kind of show who we had. First of all, Cara. I saw the vermilion flycatcher, and I thought, like, it's one of my favorite birds. This photo is not super bright, but it normally is super bright. And it really, like, as soon as she's in the room with the workshop, she's all been preparing, like, you have just this energy that comes through. And I think that's the same as the vermilion flycatcher. Then there's Roxanne. I'm not sure if she's here with us today, but she was one spreading out all the news. So you see all those seeds, as soon as you blow, like, all our information would spread out to many people. And she contacted like everyone on social media and everyone with a press release. So that's Roxanne. Thanks, Roxanne, for doing that. Here I have Jens. I obviously had to choose a fungi for Jens because he's such a fan of them, I think. And I thought this one was quite beautiful, having all the circles, and like, Jens is our cosmic network, right? So he has all the circles and is spreading the news as well in that way. And then Carlos, I saw this one, and I was just amazed by the name of it. It is a tarantula hawk wasp. Like, that's pretty much Carlos. He just knows so much about biodiversity and has so much information and knows to like combine it all. So I thought you deserved an animal that had like 3 names at the same time. I was quite impressed. And then there's me, and I was mostly like, I chose this observation because I thought this animal was just super cool, and I've never ever seen that. So if anyone has any information on this one, that is cool. So that is kind of the team that was behind this, and that has been like working to make it flow, to prepare the training sessions with all of you guys. So I want to quickly give a thanks to them. And in this slide, I would like to thank, I saw David Bain actually just joined us. Maybe you can say a quick hi. Or maybe not. David Bain is from NRS, and he's been supporting us partly financially and also getting our news a bit out. So that was a super cool input so that we could actually pull this event off. We also collaborated with Bestias del Sur Salvaje. I think that's mostly seen for the people that were in Chile, that were having an extra session with Jens and just having that time to get the Spanish-speaking people a bit more updated as well. The River Collective is behind all of this. Most of the team members are part of the River Collective as well. And that's where the event started from, right? And an obvious collaboration with iNaturalist. You've seen it all. You've been using it on your phones, so you know what iNaturalist stands for and how useful and important it was for this event. And then we were also partly supported by National Geographic Society grant. And I know many of you are actually also National Geographic explorers, so it's super cool to get that connection here as well. But now off to your guys' experiences. And a few of you have written me that they wanted to share a bit. So I'm gonna call your names out, and I hope you can, I'm gonna stop sharing my screen. So first going to Nathan Woosley.
Hello.
Are you there? Hi. Hi, Nathan. How are you doing?
I'm fine, thank you. I'm from the Chiapas River in Indonesia, in Bogor, West Java. We did 2 days on 25th and 26th, was it? Last weekend. We did two large sites. There was the upstream and the downstream. So on day one, you had team A in the downstream and team B in the upstream. And then on the second day, you switched. So we did those two areas, various sites in each area, found a lot of stuff. One thing in particular that I found interesting was this bug. I forgot what it's called, but I saw it on iNaturalist. Someone identified it for me. So that was good. It was some kind of wasp or beetle of some kind, a tiger spotted golden something. So that was, yeah, interesting. Not much of a bug person, but yeah, it was nice to see that.
Now
And
you
yeah,
are?
what?
Now you are?
Yeah, kind of. Yeah. And it was, yeah, good. I'm a herpetologist, so it was good for me to share experience. We had a lot of students from IPB University join us as well. So it was good to share knowledge, and it was good to see their enthusiasm to learn about and observe all these different animals and plants.
Cool. Cool. Thanks, Nathan. I see James already replies, and he's seen this observation as well coming through, I think, which reminds me to tell you a bit what this day is about, actually. So we now have a few stories of you guys, how you experienced the Bioblitz, and then we have indeed a few people that were like James, I think Jelena is with us, and Andrea, who are going to go into the observations that we've all done. So they're going to reflect that they've had a quick look at everything we've done worldwide. So quite excited about that. But first going to, oh yeah, in the meantime, actually, we have some breakout rooms. We tried that during the session before, so we can just have a quick conversation between smaller groups. Anna Isabel Rios.
Rios, my last name. Hi, everybody. Our river is the Rio Nazas. It's located in the northern part of Mexico. And we worked the 3 days of the Bioblitz. There were about 25 of us doing the observations, split into different teams. We liked that we had many children. I mean, it was a small group, but among the 25 of us, maybe 5 to 6 children. So it was really cool to see them, enthusiastic about finding the insects and looking at the flowers and all the biodiversity in the river, but also looking at the interactions that occur, when you look that, oh, there's an insect and then it goes a spider and eats the insect, and, or the butterfly in a certain flower, and looking at all those interactions, we think it's really cool for them to learn and appreciate all the things that occur in the river. Also, I mean, for the adults that we participated, it was really cool to share what we observed, how could we compare from last year what we saw, how it's different from this year, and also take an opportunity to look at different things. Many of us are like bird watchers, so we usually go and look at birds, but now we saw a lot of insects. So it's really nice to learn other things and look at other things. We had people from 3 years old to 68 years old participating, so it was really cool. And we are very proud what we got, so thank you.
And you have a good right to be. So that's super cool. And I like how you say, okay, we normally look to the birds, right? But now we actually also look to the insects, which are the food for the birds mostly, right? So quite essential.
Yeah. So
one big
it's
ecosystem.
Interesting. Yeah.
Cool. Thank you. Novita, Novi from Indonesia, are you there with us?
Yes. Hello, Cara, Carlos, James. Yes. Novita from this year. I lead friends from Citarum River, West Java, Indonesia. One river the most polluted in the world from last maybe 10 years. So the last visit with our group from Jakarta and around West Java region with a local community, we have some citizen science activity named Floating Tech Laboratory for 2 days. On the first day, we built like DIY tools with the children to measure the water quality and then identifying about insects, fish, and some plants at the schoolyard. But our difficulty because the signal of internet signal is very, very difficult. So we couldn't upload on the spot. And then after that, we just going to another city to upload. And then for the second day, we held some activity at
Novi, I don't know what happened. Maybe one of my co-hosts just muted you. I'm not sure.
Yes.
But we don't hear you anymore. You can repeat
Yes.
your last little bit? Yeah,
Okay.
we hear you again.
Could you hear my voice? Okay. Then for the second day, hello? Yes, for the second day, we held for next activity with a local community for citizen science at the downstream area near Jakarta Coastal Sea in the mangrove forest to find some like fish like giant mudskipper and then long-tailed monkey and so many mangrove plants, also so many birds there. We identifying that the fish could live better in sedimentation water because the fish like bioindicators that the water contain heavy metal. So the researcher will identifying more in the laboratory after that. So maybe for the next month, we will get the data and then about the parameters. Yes.
Cool, yeah. I'm happy you shared your experience because I was like on the Instagram trying to reshare all the stories that came in.
Yes.
And your lab just came up 100 times, I think. Like many people were sharing it and many people were so excited. So I was like,
yes.
what is this actually really about? So
yes.
Super cool.
But Cara, I think for this year's bio, not so many can upload the photos because our difficulty because the signal of internet there. It's very rural area because we just use iNaturalist just so difficult, so the parties couldn't identifying more in a naturalist, but we get so many photos, then I'll upload later in our drive.
Cool. Thanks, Novi. That's amazing.
Yes, thank you, Cara. Also, Carlos, thank you.
Cool. Aurora, are you with us? Don't see her. All right, let's then go to Anya.
Hi, everyone. Well, our river was Ahuacapan. This river is a mountain river. In the river, there's an influence of natural reserve. So the river that we went were really conserved because that river provide water to a town named Ahuacapan. So we have the fortune to know a conserved river. And this type of river is in a mountain high. So we have to climb and do some walk. So in this event, there were 23 participants, more of them students and professors. We have three professors. One were expert of birds, another one of flora, biodiversity, and Dr. Claudia, expert on water quality. So while we were like climbing, we were seeing the flora part, the riparian zones. So Dr. Arturo Solis helped us to share his knowledge about the flora. And then we saw some birds. So Dr. Sarai shared his experience about the birds in the local community. And then we went to a pool where there were a cascade, and we have a chance to know more about water quality with physicochemical parameters, and then we know a little bit more about microinvertebrates. So the participants were so excited because the students were from different disciplines, and some of them travel from 3 hours from another city. They were so interesting, and they were like knowing about the biodiversity, where the areas were conserved, because some of them came from a city. So the kind of river, it's like in a canal. So it's very, very like contrasting the type of river landscape that they meet. And one species that I personally was very interesting on was this butterfly that has in its wings 88. It was cool seeing that butterfly. So that's it.
What did you mean with the butterfly that had its wings?
Yes, in its wings it has a figure that it's like 88 number. Yeah. It's really cool. Yes.
That's beautiful. Cool, thanks Anya. And amazing to hear that you managed to, through the Bioblitz, just bring different disciplines together, right? I think that's really cool, because people tend to start to focus, and it's a pity because it's all connected. Yeah, cool. Thanks for your experience. Natalie just mentioned that she also actually would like to share a little bit. I'm super happy to hear of it.
Yeah, so I had my Bioblitz in Bannockburn in Scotland, at a burn, not a river, but we connect to the river. And yeah, it went really, really well. It was an open event, so we didn't know how many people were going to come. This was advertised like in local schools and just for the general community. And so yeah, this is something to learn from, I think, because I didn't know at all how many people were going to come, but we had over 100 people come and attend the workshop. So we had a river dipping workshop that ran twice and was like full, like it was so busy. And the best part was seeing all the kids in the burn, like just playing and using the samples and finding the bugs. And I think this was the best part. And then we also had a pollinator workshop, and we had a mindful moment in nature by the burn. So people were connecting with the burn in different ways. And older people in the community said that people haven't been in the burn like this since like the 1960s, where children would go and play and by themselves, like in a big group, and feel safe and just like experience being in nature rather than being inside. So I think this is some of the best feedback we've had, and everyone is asking for it to be every year. And I think explaining to people that this is something that is happening all over the world really help people to get excited about it. And I think I just need to figure out how to combine the workshops with actually maybe to do a workshop in advance to show people how to use the iNaturalist app and things. That I was counting on this during the day and it didn't always work, but yeah, it was a great success. I'm very happy.
Cool, thanks, Natalie. I got excited all the time that I saw messages from you. It was like, wow, what they're doing there. And it's not too bad to not expect that like you got all the observations in, because what you did was super ambitious, I think, combining it with all the workshops, and then you had this beautiful map and so many kids around. So I think like super inspiring, and I think we can all learn a lot from how you approached all this. And your feedback with this like historic connection to the river, amazing, super cool. Is there anyone else that would like to just share a few words since we're here about their biodiversity? We're gonna go quickly into breakout rooms for like 5 minutes to just have a bit of a chat going on between smaller groups, so everyone can chat a little bit. So maybe we'll just do that unless somebody really says like, I want to say something to everyone now. Okay, then I'll quickly set up breakout rooms. 3 to 4 people. And like my suggestion, if you get into the breakout room, is to talk a little bit about like one thing that really surprised you during the day. For me, that was, for instance, like we were doing this Bioblitz on a little creek going like at the side of a river. So we were rafting a bit, and then we walked up this little creek onto a waterfall. And there was just this cascading waterfall, and we went behind it, and we had this biologist and like bird watching person that knew everything about birds, right? And we came into this cave, and I was like joking to him, I guess you won't find any birds here, or do you see birds? And he just looked up, and there were like 2 white-collared swifts in there, just hanging there waiting for us, and they were making sound as well. And I was like, amazing, like you just walk into this dark space and he just still finds a bird. So that was really, really cool for me. So something like that, just share it around. And if there's one thing that you would like give as the advice, if there's something that worked just really well for you, for instance, making this map that Natalie did so the children could go by, or you also had this bingo card, right? Like you had a bingo of places to go through Bioblitz, things like, okay, try to look under a stone, try to look in a tree, try to, you know, something that worked really well for you that you could give as a tip to the other person. So that's for the breakout rooms, and we'll get back in like 5 to 7 minutes or something. Cool, have fun.
I am back in the UK, yeah. Coming into an environmental conference after flying to France for 3 days. I apologize, everybody. But yeah, my first trip out of the UK for 2 years, back in the UK now, and preparing for our next season, Vera.
Amazing. David is the guy behind NRS that has decided to support us quite a bit, not only financially, but also in spreading the news. So thanks a lot for that, David.
Yeah, no problem. It's great to see so many faces on here, and I guess it's interesting. This community is obviously very much from the scientific front, and I think I come from the other side of that. I just said in the breakout room, you know, every time we go kayaking, you could argue that it's actually a negative impact on the river. You know, the river flows with or without kayakers. So I think it's important that, you know, a sport like kayaking and NRS can help support the scientific protection of rivers, because you guys are the clever ones that are actually thinking about this up here, rather than the shouting about how much you care about rivers. So yeah, thank you for inviting me on the call.
Cool, thanks, David. And obviously for many of us, like the same passion comes through science, through kayaking, through anything, but it's a passion for rivers, I think, that we all share. Cool, we're gonna go to Sandra, who also wants to share a bit about their experience. And let me know if you can share your screen, or if I should have done something for that. If not, I can make you a co-host.
Thanks, Vera, and thanks, Jens and Carlos. It was a really incredible experience, and I will share a small presentation to show you our experience in Mapocho River in Chile. Wait, let me know if you can see.
Yes, we can see.
Yes, it's coming.
And I need the presentation. Okay. It's okay?
Yes.
Okay.
It's okay.
And the first thing I would like to say is that Bioblitz experience in Mapocho River, it was inscribed in what it formed part of another research from Universidad Católica of Chile. That Jens is part of this experience, and we had a very big call in Chile, a very good, a very incredible response from the people. We had 4 or 3 days before calling to the people by different media, and we had the first goal that it was incredible that we connect with 12 ONG or NGO organization, environment organization from Chile, that upstream and downstream. And it was incredible to connect with this kind of organization because we had a lot of time to recover in the river, but alone. And this event, it was a good, ¿no es cierto, place to connect with him. And the other thing that we have sponsor, like you see, Ministerio of Cultura, municipios, 9 boroughs from Chile, Ladera Sur, and other media platform. And we involved with biologists and ecologists and science people that work alone in different universities, for example. And from this event, we connect with that people. It was incredible for the experience that they have. Wait. And that is a kind of situation and the experience of in 9 points along the river, in 9 boroughs, in different places, like Barnechea, Vitacura, in upstream and downstream from Pudahuel, Maipú, Padre Hurtado, Peñaflor, Talagante, El Monte, Isla de Maipo. The people came from different places in Santiago, and sometimes they don't know these kind of places because they are boroughs, peri-urban boroughs and rural boroughs that the people don't know from Santiago, from the urban areas. And the other thing that we did, it was a physical map that it was a very good connection with the value of the river from the species, vegetation, etc. And it was good to connect, for example, kids, not only for the people that know the iNaturalist platform. It was a very good increase for the value of the river, to know the value of the river. And here you have the organization that that was involved with the experience, and we are working with him in another kind of experience for the next future. We have more than 1,000 observations with the iNaturalist platform, more than 300 people that participate in this experience with Mapocho River, and 235 species identified with the people. And we were talking with Jens that this experience is not only 2 days, or it's not finished with the 2 days. And I think that is a very good point to increase the experience in the next time with another kind of research, investigation, or experience. And for us, it will, it's a very good, for example, situation for increase the knowing of the people to start this kind of project or specific project along the river, for example, for the borough. And we are working with iNaturalist for the next time. We think it will be a very good experience to continue not to finish with the iNaturalist. And we have a next activity in January. We are planning in relation with this BioBlitz. It will be the Mapocho 42 Kids. We did the next year, and I think that's our experience. And if you have any question, please let me know. That is, Jens, if you want to increase my speed, maybe it not was to be clear, but if you need to.
I think that was perfect. Yeah, it was really nice.
But maybe Jens, you can say a little bit, because this is super inspiring, right? Like we started the BioBlitz year. Oh, sorry. Jens, slow. Go ahead.
No, sorry, my internet is too slow, so I'm like 1 or 2 seconds behind everything you are saying. No, let's talk about that at the end of this session on like this next step on like using iNaturalist as environmental monitoring tool for environmental movements. But now basically we will have one more story which Carlos will show, which is our story in Thailand.
Yeah, Aom has just surgery with her dentist, so she might not be joining, but she sent this beautiful video. I'm gonna share my screen very quickly. And here it is. I hope, I'm not sure if sharing with sound. Share screen, sharing with sound. Now I got it. Should be working.
On World Rivers Day, we survey biodiversity around the Moon River, where 25 roots were discovered 100 years ago and protected as a monument. We explore along the Moon River with Tha Chang Raj Bamrung School and Kora Geopark. We were excited to see many examples of local fauna, such as 12-inch earthworms, beautiful dragonflies, and a skink. We also enjoy seeing local flora such as passion flower, snapdragon root, and gooseberry. The day before, we did the BioBlitz at Moon River tributary, Lam Takong, with Sungnar School, Geopark staff, and volunteers. At this site, it was this student's first time seeing a spiny eels. We asked fishermen to show us what they caught. We observed insects, birds, and even a venomous green pit viper. Bamboo, fig tree, and monkey ladder are common around here, as well as mushroom and lichens. Through the lens, we observe diversity. Through the heart, we feel the connection of life, people, and our rivers.
That was an amazing experience, and I'm sure that many of us share pretty much the same. So this is like a global community now.
Wow, so cool. Both this and the story of Sandra, right? Like, to be honest, we started this BioBlitz last year when we were all a bit inside and half depressed with the pandemic, right? As a just reason to get out. And then seeing it within a year, getting like stepping up and people really getting a lot of organizations and people and heart behind it. It's really amazing to see and inspiring. So we'll definitely do a BioBlitz next year again. Now we want to actually, as David also said, like the science behind it can be really, really helpful and can be really a monitoring tool. We'll talk about it a bit later, how we can use that a bit more. But to just have a bit of a glimpse into what we actually observed, we have a few people that dove into the observations a bit and can have a bit of a talk about it. Jens, do you have any? Okay, Jens is out. I think I'm gonna just give the word maybe first to Jelena. You know Jelena from the amazing presentation she did to teach us a bit how to do bird watching. And now she's been diving into the birds, I guess.
Okay, hi everybody. I'm glad that I'm here again with you, and I really enjoyed today just scrolling endlessly through all the birds that you observed, which was really cool. I was marking like places where I really had to go for some serious bird watching when I have time and money. Okay, I will share my screen now. Okay. Okay, so I hope you all see my screen, right? Cool. Yeah, so I'm Jelena and I study in Munich with Jens, or we were studying together, soon finishing. And I really enjoy bird watching as my hobby, and I also do some other behavioral things, maybe professionally. But I really, as I said, enjoyed this really cool observations. I did this the last year as well, and I have some memories from last year observations. So for example, this black phoebe here was the most observed bird this year, and then the snowy egret, pretty much a lot, and this really cool spotted sandpiper. But this bird was on the 4th place, this vermilion flycatcher, which was like the most observed bird last year. So not many things, I think, changed in these observations numbers. This was everything that I had to go through to see and to choose the very pretty birds that I have to, like, it was really tough. Today I really had a tough day. I was like, oh, what am I gonna say? Which species am I gonna choose? But overall we had 237 species this year, which is almost 10% of all the species that you guys recorded, which I think is pretty small worldwide. So not a lot of birds here, but still better than the last year. Overall, probably not a significant difference because this year there was more observations overall. So I'm really, so the numbers are pretty much the same. And yeah, also, I was kind of comparing some spots from this year and last year, and I must say that it is very interesting to look at these things. For example, this La Meraviglia del Tagliamento theme from Italy last year had 91 bird species, but this year they have only 12 species of birds, with this very cool observations, of course, here of the stone curlew. But here, this is the example actually how things can change when you have certain people in the team or when you don't. For example, that's one factor to take into the account when using this data for any comparisons, because there is really this kind of bias sometimes in species, which you can see here, that last year they were really concentrated on bird watching. This year that was like not an objective because maybe they lacked an expert or just there were not so many birds. But similarly as last year, and even better, there was like Rio Nazos in Mexico. I really enjoyed it. And so they had last year 44 species, this year 54, which is good. There was like this 10 species that actually added, I think, to the last year. So it was cool. And yeah, so also what I saw just now is that some observations should also be checked twice. And I think that's a good kind of thing to do when you are uploading something and letting a lot of people identify this. Because this is India here, and this is this dam where people in India were doing observations, as I understood. And this is a feather of a probably egret bird. However, somebody said that this is a willow ptarmigan, which looks like this, and it cannot be in India. So it's always really important when you upload and when you see people commenting on this, maybe like just check for the distribution, because some things are just impossible. Like this is not possible. Okay, so then let's start with some beautiful birds. There were some really cool sandpipers, and I said already that this spotted sandpiper is really the most dominant, mostly observed one, but a killdeer is really cool. I think this is one of the biggest sandpipers, and he's really like, they are crazy birds. Their display is crazy. They just pop their eggs wherever they see, on a lawn, in front of your house, in your garage, they don't care. And also they were like the smallest sandpipers, which is Calidris minutila. Super small, tiny birds, which breed in the tundras in Northern America, I think, and then they, I think, migrate to Mexico for wintering in wintering grounds. And this was actually recorded in Mexico in Rio Nazas. I think this one observation was there, or two, but that's a cool thing that this species is observed there. Then we have some pretty much common river species, and maybe the most favorite of all, such as these kingfishers, this green kingfisher here, then these two really similar ones, the pelted kingfisher, like this one here, and then the ringed kingfisher, this one here, and the European one without a crest here. So they were also recorded, which is really cool because rivers are, I think kingfishers are kind of like marks of a river, and a nice and healthy river. Then we had the same species like last year, and there was this one, Loggerhead shrike, which is near threatened. This was also recorded in Mexico with 2 observations, and I think on a similar spot as last year. I need to double check that, which is cool to see some repetition in these observations over years. Then, unfortunately, this year we didn't see this really cool Juan Fernandez Firecrown from Chile, which I was like really expecting. Last year, I was super excited about seeing this bird, and then this year I first searched for this, but unfortunately it wasn't seen, which is again either due to the not being there or just some other factors. Okay, then again some really interesting observations, such as cattle egrets. These birds are really interesting. They're also like monophyletic, which means that this is the only species from this Bubulcus genus. They have a really wide distribution. They're also in Europe and Americas. And although none of these birds are really marked as endangered, I think that on global level, maybe they are not, but locally their populations must be decreasing. Then there was this Vanellus indicus, very cute lapwing. Also some swifts, this one Apus affinis, and I also saw the one that you talked about, Vera, from the cave, and this bulbuls, and these drongos, which are so cool. This bird is such a manipulative, strategically manipulative bird. I don't know if you guys saw ever video of how they trick meerkats to eat their food, but they literally first imitate the call of the predator, then they imitate the meerkat call and trick them multiple times and gain their food, which I think is cool, but for meerkats. Then we had some hummingbirds also. So for example, this black-chinned hummingbird, ruby-throated, and giant hummingbird. I was really excited to see how giant is this giant hummingbird, and apparently it's the size of a European starling, which is starling size. I don't know, that's my reference. you gotta find your own, but yeah, so it's apparently the biggest hummingbird, and but it is lighter because it's so slender and with this nice slender beak. Then also we had some birds of prey, which are really cool. I think this one is from Chile, black-chested buzzard eagle. Then also this variable hawk. This bird here is very polymorphic, which means that they occur in so many morphs that I think they have like around 27 morphs, which means that they look completely different all the time. It's similar with the common buzzards in Europe. They also can have different morphs and different colorations, and even males and females have that. So that's pretty cool. Also this eagle, which I've never heard before about, and now I did. It's a black eagle, usually in Asia, common. Then the smallest falcon, Merlin, Falca columbarius. It's really the cutest, smallest falcon bird. I don't know. And then it can also kill cute prey. And we had also, for example, the short-sawed snake eagle, which are also kind of declining, although they are not really like threatened. But yeah, we had a couple of observations of them, and a cute Oriental honey buzzard. These buzzards are I mean, you can see here, like they are actually not related to buzzards so much, but anyway, you can recognize them by their head looking so much like a pigeon. And they have this cool display to females where they have this like roller coaster where they glide and then they go up. Yeah, I think all of the Pernis species. Yeah, so it was similar like last year. I expected more bird observations. Honestly, there's so many birds in the world, but most birds observations were within a couple of transects again. And usually it was the mostly common species for that area, which is also fine. And that is also one of the things when you go to these places, when you visit one river over years, you will be used to and you will know what to expect there. And you will, of course, like first and mostly record the common species. Also, I think that bird watching in big groups is always not a good idea or such a good idea. It is hard to do it like that. It can be fun, but it is definitely not fun for birds because some of them are pretty timid, but some of them are like, nope, I'm not going out. Also, the terrains and habitats are different because a lot of us have also observation throughout the city. I mean, in the city of Munich, I was pretty surprised this year. There was pretty much a lot of birds, but then also there is a limited number of what you can see. So there's some peak, and it depends, right, where you are, in which moment, at which time of the day, and yeah, also quality equipment is needed to kind of see and record the species. And if you don't have an expert in your team, which also probably has these things, then it's also not fine. You cannot see them. But overall, it was really cool. I really love watching, like just scrolling through the iNaturalist. And yeah, I think that's what I have to share with you. I hope this wasn't boring. I hope that I can answer any questions if you have. And yeah, thank you very much for listening.
Jelena, hi, I'm Carlos.
Hi.
Is there any endangered or IUCN species that you managed to find?
So this, IUCN also classifies their species endangered by a global distribution, and they use specific, and like they really are, when they classify an endangered species, they have this range of like a global range, right? So most of these species would be IUCN not endangered. However, I feel and I think that that's not something that should be looked on a global level, but rather local, or like on a continent or something, because I think that, for example, these Sandpipers, this small one that I found, it breeds in Canada, and then I went to some Canadian websites where they estimated like that that they really are putting, the attention to conserve the species and take care of it because it could be declining, and most of those have stable populations. But these shrikes, for example, the shrikes that I showed, it was kind of near threatened. So when you go to IUCN and type this some of these birds, most of them will be not least concerned. Yeah.
Okay, thank you.
Yeah.
So now we will have Andi after this amazing presentation of Jelena about the birds. Andi will talk about plants. I don't know if also you consider it lichens or mushrooms or fungi, but you will show us now what you got. Thank you, Jelena. That was amazing. Can you share screen, Andi?
Yeah. Okay, let's see. All right, can you guys see it okay?
Yep, totally.
Great. Okay, yeah, hi, I'm Andi, also living in Munich and studied with Jens at the LMU. and I'm a botanist and mycologist. And it was really interesting to look through all of your photos from your different bioblitzes and get to do a short review of the cool plants and fungi that I saw. So I'm going to start with just a quick overview of all of the plants and fungi that we saw compared to what we saw last year, just some numbers, and then I'll get into the good pictures. So out of the over 2,000 observations of species that we saw, the majority of them are from plants. And actually, what's interesting is there were more observations of plants, but it was a smaller percentage of the total observations. So it seems like people who were observing maybe insects and birds took a larger percentage of the observations. But that's really cool because that means like we had way more total observations. And the same thing with the fungi is there were more total observations from last year, but it was like a smaller percentage. So it seems like the animal people really kicked up their numbers there. Similarly to what Jelena said is that I saw quite a few misidentifications. And so if people who are really like familiar with their local plants and fungi could go through some of these observations and like double check the names and make sure that they're accurate, because there were a number of misidentifications. But this happens with citizen science people who aren't trained necessarily. And with the iNaturalist app, you can have it suggest something, but like not always the first suggestion is correct. But if you're not trained, it's hard to know exactly what you're looking at. So, but for those of you who do know, maybe you could help us out there. Okay, so we had observations of plants in every place that we had a BioBlitz. For obvious reasons, because they're easy to find and take pictures of. They don't move around too much. With the really large numbers coming from Chile and India and the Indonesian bioblitzes. And there were so many beautiful flowers that I just wanted to do a quick like screen capture of a few of them. And what I thought was really interesting is that there were so many plants that were considered introduced species that were observed, and so few that were considered threatened or endangered. And that sort of like leads to, or not lead, it sort of helps illustrate the point that introduced species are sort of taking over these wild spaces and causing problems for more endangered and threatened plants, like taking over habitat. Our most observed plants were two flowering plants, and one of which is the Eschscholzia californica, which actually is introduced in the location of Chile where it was most observed. And also the castor bean, which also was introduced into Central and Southern America. So I think that it's interesting that both of the most observed species were considered introduced species. And then I just wanted to take a quick run through a couple of the like pretty flowers with some photographs that were taken during the bioblitzes. So we have this Boheravia,
still not reading.
which was from Mexico. We had the Puya, which was from Chile. I really wish this one was flowering because it makes these really beautiful blue flowers on this spike here. And then we had this Tropiolon, which is really, really nice, making these little shooting star flowers. I really want to see this in real life one day. And then what I thought was so neat is that we had observations of a Marcantia, which is a liverwort. These are tiny little guys, which are not truly vascular plants and are very, very commonly overlooked. And so it's so cool that we had some observations, and specifically this one with this little palm tree looking structure is actually its sexual structure, and those are so transitory that like it's really hard to find them. And so I think this is a very, very cool shot of this little guy. Okay, and then I'll just move on to some of the fungi that we're seeing. And again, actually, like fungi were documented in almost every bioblitz that happened, which is really cool. And our most observed fungi were the Milky Koncap from India, and this little guy who I can never pronounce his name right, but what was really cool is he was also observed in last year's bioblitzes in Chile, and so it was really nice that we got to see him again. And so it's like the progression where it starts off as this white structure, and then it moves into this sort of orangish color as it matures, and people were able to find both colorations during our bioblitz past weeks. And then just some really cool fungi that we're seeing is this stinkhorn from Colombia, which wow, just one of the most amazing fungi that I've seen. And then this little guy, he's a very, very small fungi who does the bioluminescence, which not very many fungi do, but when you can see it, it's so, so cool. So whoever got this shot, really credit to you, because that's a great, great shot. And knowing that you could get it to bioluminesce. So thank you for that. And then finally, near to my heart are the lichens. And it was really cool to see people taking an interest and photographing or attempting to photograph lichens and identify them, because they're difficult and oh, don't pay attention to this top part, that's wrong. But anyways, so yeah, we had a couple of shots of lichens, which Cladonia being one of my favorites. It was really nice to see other people taking an interest in those. Yeah, so thank you for paying attention and participating in the bioblitzes. And as a scientist, I just want to say, like, really, really thank you so much for documenting these things. In my master's thesis, I actually was able to download and use some data for the like global locations of the species that I was looking at, and it's such a great tool, and getting people involved is such a cool thing. So thank you so much.
Wow, thank you, Andi.
Thank you, Andi. Go for it, Vera.
Yeah, that's amazing to hear that, like, because I think many of us are actually not a real scientist. I'm, at least myself, not a biologist, right? So hearing back from you that you actually use it, also the biologist that came with us, he was actually using iNaturalist data for his papers, right? So that's really encouraging to know that we're not just doing it for fun, right? And super cool to see you, both you and Jelena, just putting that much passion and excitement into going through all the observations and actually being happy to do it. So super cool. Yeah, I think last we still have James to go, right? Sorry, I was gone for a bit because internet kicked me out. But I think James also prepared some presentation and went over all the insects, no? Okay. James doesn't seem to be here.
Can you hear me now?
Still me? Ah, there he is. There he is.
All right.
Amazing.
Yeah, I did. Let me share my screen.
Also, James did a bit of an introduction for us a few weeks ago. explaining us where we could look. And super cool that you're also diving into them now.
Yeah,
thanks.
So unfortunately, I wasn't able to take part in a BioBlitz because I was flying from California to Chile, where I'm going to be working for the next year and a half. But I, like the other two people before me, had a ton of fun looking through these observations because there were some really cool insects. So to start, just a quick breakdown of the data. Behind plants, insects were the next most commonly recorded group, which is probably as it should be if we're looking at things on a global scale. We got 775 total species, which was about 32% of the total species recorded. And there was a ton of really great diversity. So I had a lot of trouble choosing which things to highlight, so I just ended up going with the things that were most commonly observed. So here are the top 6. Which are in the top left, the plain tiger butterfly, then the scarlet skimmer, the mottled emigrant, a bitch jewel, which is a very fitting name, it's just gorgeous, the yellow feather legs, as you can see, it's got some very yellow legs, and the common Mormon swallowtail, which swallowtails you can tell by this little lobe on the end of their wings. So originally I was going to show the distributions of these species, but when I started to go into it, I realized that these top 4 and this 6th one were all from our very active group in India, while this one was found in Indonesia, and then also one observation in Vietnam, which was pretty cool to see that actually spread across 2 different countries. So that kind of quickly brings me to sampling bias, which the one I was just talking about is sampling effort. So just because 5 of the top 6 were from India does not mean that there are more insects in India than anywhere else in the world. It just means we had more people out there taking pictures and uploading them. It's just purely a function of that. And if you noticed, those top 6, 3 of them were dragonflies and 3 were butterflies. What do those have in common? They're big, they're colorful, they're pretty, and they fly around a lot. So it's really easy to see them. So if we were accurately representing the insects that were there, it would probably be a bunch of really tiny brown beetles and moths, but people aren't really looking for those, so we didn't get many observations. And this is just a funny cartoon. So I put together after that just kind of some more of the most commonly seen groups. So some other Lepidoptera, which are the butterflies and moths. These are the next 4 most common. A lime swallowtail, which just the patterns blow my mind every time I look at it. This is an Empress Layla, which is actually a moth, but it's really gorgeous. A common tiger, which is closely related to the most common species that we saw, the plain tiger. And then a bordered patch right here. Next, we have the other Odonata, which are the dragons and damselflies. And there were probably 15 or 20 that all had 10 or more observations because they're just so, like I said, they're so colorful, they're so beautiful that they really draw your eye when you see one and people want to take a photo. So we have the pink jewel, the 3-striped blue dart, the marsh dart, and the dusky dancer. And even just in these 4, you can see kind of the variation in color between all of them. They're all really attractive in their own way, but they all have such like differences in terms of body thickness and where they have their wings and colors and all that good stuff. So then my personal group of research is the beetles or Coleoptera. And so I decided to highlight the 4 most commonly observed beetles. And here you'll actually notice that the top 2, Platycorynus peregrinus and Monoleptula signata, they don't even have a common name. That's just kind of a function of how many beetles there are out there. There's just simply not enough knowledge about them to even have something in our common description. But as we can kind of see with all 4 of these guys, they're all, again, they've got stuff that draws your eye to them. This guy's shiny. This guy's got these bright white spots. This is the golden spotted tiger beetle that I can't remember who mentioned earlier, but someone mentioned this as one of their favorite observations, and I agree. They're just incredible. These guys are pretty much top predators. These are like the cheetahs of the insect world, super fast, they'll run down anything. And then someone else, I think there at the start, you were saying that you identified yourself as this Asian spotted tortoise beetle, which I just love both the shape and the transparency. This whole group is all kind of shaped like this, and they can have a crazy range of colors. And they're also closely related to another group called UFO beetles, because they kind of look like flying saucers. And then after that, I highlighted a couple more that were not from the same order, but these were kind of the miscellaneous ones that were observed most frequently. We have a very colorful grasshopper up here, the Carianda spuria. And then we have the lychee shield bug, which is again, super colorful, really catches your eye. So these are in the orders Orthoptera and Hemiptera. We have a mantis down here, probably one of the bigger insects that anyone was running across. And these guys are, again, really top predators, but they're ambush predators. They'll just hang out, wait for something to come by and snag it with these big claws at the front. And then this is a Tractomorpha alata, and this just kind of, to me, highlights the incredible diversity of insects, because this and the one in the top left, these are both grasshoppers, but look at how different they are. This guy, totally flat, he doesn't have nearly as distinctive colors, or the legs really sticking up, they kind of blend into the body, have this long frontal mouth part. It's incredible that things that are so different can actually be relatively closely related. And that's really everything I have. Just a quick plug for the work I'm going to be doing. These are some photos taken by my mentor who works with National Geographic, and I'll be posting photos that he and I take on this Instagram handle. And if you ever want to contact me, I've got my email down here at the bottom. And that's everything. Thanks.
Amazing, James. This is super cool. Thanks for getting in that insect that I identified myself with. That was cool to learn a bit more about it. Honestly, the three of you, I think it's amazing. Like, I've planned and wanted to go through all the observations and kind of get a clue of what everybody's been doing around the world. But for me, it's kind of an impossible task to make some sense out of it. And then you guys just go into it and conclude everything and kind of wrap it up into one presentation of 5 to 10 minutes. I'm really impressed, and it's such an addition to this session, I think, that you guys have done that. So thanks a lot, all of the three of you, and all of you pointing out how potentially biased and how still there's a challenge with citizen science, right? Does it really represent what we see? Does it really I think, Jelena, I feel you're underrepresented with the birds, only 10%, right? But how difficult is it to actually get a photo of a bird? It will fly away by the time that, I think there's definitely some challenges in using this as science. And I think that's one thing that we would really like to look into a bit more for next year. So I think this year we've been focusing a bit on the storytelling part behind it, which we find super important. And I think that should remain core. Right, so how can you use a day like this? How can an event, what's your reasoning behind it? What's your motivation? And really bring that out as well. And not only stick to the science, but I think for next year we would kind of want to explore a bit more okay, what can be the science behind it, and how can we make it easier for people to make something that's valuable for them as river protectors as well. So how can you monitor in a way that you get valuable data? So if there's people in this chat room now that are okay, I would really like to think along with that, please write us an email, and then maybe in a few months we can set up a meeting with those people that are really into that, and that would just really like to think a bit okay, how can we make this more science-based, right? Higher value for science, so that next year we can even have a bigger impact besides the stories. Which also leads me to go to Jens. I don't know how your connection is at the moment, if you can say a bit about this last meeting we're hoping to have.
Yeah, I hope my connection works and you can hear me on time. Well, basically, like Vera said, we plan on doing one session with Carlos, especially aimed for those who want to keep doing environmental monitoring at their own home rivers and using this as a tool, for example, to protect and to fight against the construction of either project or the channelization of the river or the pollution of the river or any other way of human impact into the river. So we plan on making this session aim, for example, to build up a project for your own river that's not only for this particular day, but you can keep track of the observations during a whole year, maybe during 5 years, or maybe comparing some taxas in time. So the idea will be going a bit deeper into how to use this tool also for actual river protection, not only raising awareness, but also like having some actual useful data for the river defense. Yeah, we are looking forward to that session as well. But as like Vera said, this is our main aim. It's mixing this storytelling, this idea of raising awareness and also of connecting people to rivers, but also like making use of this data that we are getting each year, and each year even more during the BioBlitzes. But yeah, thank you, James, Andi, and Jelena for those sweet and short presentations, making sense, like Vera said, out of so much data. In the end, the data is only numbers for actual species, and that's also important to remind. And also, thank you all for telling your stories. I think that's actually, for me at least, that's my own biggest interest, to see how these activities may connect us all around the world, like making this global community that fights for rivers. There are many platforms, but this may be one of them. And also Carlos all the time, and Vera, Carlos, you have been like being so important to solving any doubt concerning the iNaturalist app and making way more than you are supposed to do. And also Vera and Cara, we have been kind of trying to be a bit aware of everything that's happening, but it's just too much and it's really beautiful. I don't know if you have any other thing to say, Vera, or if anybody has anything else to comment, but we are really looking forward for the next year. But also stay in tune if you want to participate in the organization. You are welcome to approach us, or also if you're interested in this workshop, we will anyway we will post it in the WhatsApp group, or also share it in an email around. So yeah, Vera, Ricardo.
Yeah, one practical note, I'll be sending an email out a bit later with a little short questionnaire, just to have a bit of feedback from you guys, and feel free to write as much as you want, but also just to fill it in quickly. It would be really valuable for us to have that. So that will come in soon. And no, biggest thing I want to finish off with indeed is a huge thank you, and a huge super cool to see this feeling of collaboration around the world. And thank you, Jens, thank you, Carlos, thank you, Cara, thank you, Roxanne, who's not here at the moment, but still, thanks everyone who contributed to this, and all the stories, all this motivation coming through the chat as well, just keep motivating us. So it's amazing.
One more thing I forgot to mention, we are having a video for the BioBlitz like last year, where we had some interviews and we show also some rivers from all around the world. So please remember, we share with you this folder where you can upload your videos, so we can also show some shots of your rivers. And also, if you are interested in the storytelling part of this, there's already this story map possibility that some of you I already know are working on that. For example, I think Anya is also working in her own story map to show in your own community, or also to a broader audience, what you were doing during that day, or before and after. So you engage more people into the river, not only the participants, but you can share also that story map then among your peers, people interested, maybe even media, so to raise more awareness about the problems that your own river is encountering. Well, yeah, thank you a lot. And again,
maybe we just finish up with one. Okay.
Go for it.
I was going to say, maybe we just finish up with one, like Carlos just sent a photo, but everyone at once turn on their cameras and just like have one last wave.
Excuse me, I just before the photo, or maybe after, I don't know, if I could say just a little words.
Yes, please.
Of course, Rodrigo, say
Thank you. Well, hi, I'm Rodrigo from, well, I live in Santiago, but along Marcia Tomasoni, we worked in this year Home River Bioblitz in the Aconcagua River, in the Aconcagua mouth, la desembocadura of the river in the Humedal de Concón. And to be part of this group, it has been very inspiring. Also today's talks. And we would really appreciate to keep communicating with this global community and also with the Chilean community. We would like to repeat the experience of the Mapocho River. We would like to work along the whole basin of the Aconcagua River. So I really expect to keep talking with you all and only say that thanks a lot. It has been great to be part of today and all the groups. So that's all. Thank you all. And let's go to the picture, I guess.
Yeah, let's
Cool.
Thank you, Rodrigo. Those are
turn on your
cool
cameras,
parts.
please.
You'll have to take it, Carlos, because I am on my phone and it just doesn't work. Yeah, smile.
We should have done this at the beginning of the session when we were more, but it's also okay.
That's okay. I got it. I got it.
Yeah, we forgot that for sure. Yeah, but it's cool. Thank you a lot. And really nice. Yeah, thank you. Looking forward to see you also in that session.
Keep using the
Over.
WhatsApp chat. Go for it. Keep using it. Keep connecting. Sending us photo of weird insects and stuff like that.